Well, this is one of those rare opportunities where David and I get a chance to sit down and just sort of chat about what's going on and let you guys in on some of the excitement around AGT. And I got to tell you that it's already been an exciting year. We're only in early February right now, but the HIV clinical trials is so close you can taste it. And there's a huge amount of excitement, anticipation, and a huge amount of work around the company. So I borrowed David briefly because I just feel like it would be exciting for you to hear about it. But he has very little time, and he'll be running right back to that right after this meeting, I'm certain. Without further ado, I guess everybody knows David Pauza by now, the scientific genius behind the company. And he's not alone in the R&D department. He has a fabulous team working with him. He's done a great job recruiting some top-notch people. They're all working incredibly hard in order to achieve milestones this year and get us through to the HIV functional cure. But from 30,000 feet, I wanted to give David an opportunity, and really you guys an opportunity to hear about the progress on the way to the clinic. So David, can you kind of tell us where we're at and how things are going?
Sure, thanks Jeff. I think it's really important, and it feels great around here because a lot of things that weren't very clear or kind of fuzzy related to the HIV project are now becoming very clear and very precise. And the most important thing in that category was our ability to make a cell product for treating people with HIV and to manufacture this at a clinical scale so that we can actually do it in the correct way, do it in a way that'll be acceptable to the FDA and safe for the patients, and move this into a clinical trial starting this summer. And we faced a tremendous number of obstacles in developing this cell process. All these things were due to the very unique aspects of people with HIV. Even if they're on therapy for a long time, they still have unique characteristics that we had to combat. And it was a full court press of the whole team, trying to be creative, trying to look at alternate procedures, and then adapting these procedures so they worked in the hands of our commercial manufacturers. It's one thing for us to get things done here in the house where we have the most talented people you can find. It's another thing to transfer it to a commercial manufacturing slot where we have to take whoever they hire and make this thing rock solid so it can be done repeatedly. So it's extremely exciting that that's done. We call that locking down the process. That means that we're not going to deviate from that process now. We think it works in a high enough proportion of people with HIV, and it works reproducibly, that we've got the bugs ironed out. And having the bugs ironed out makes Jeff really happy, because instead of seeing months tick away in things that don't result in a date for starting the clinical trial, now we pretty much know how everything maps down to the minute between now and the IND submission.
Wow. Terrific.
And so that's the update on the HIV project.
Well, first let me say, and I just want to iterate the point that you made, is that it was a tremendous accomplishment to be able to automate this cell processing protocol so that it was scalable and something that we could distribute across the world eventually. This is going to be very important to the commercialization of this. And you guys didn't just try to be creative, you guys were creative, and you solved an immense number of problems. And I actually saw along the way that you were kind of training Miltenyi on how Miltenyi machines work from time to time, and actually showing them that those machines have more capabilities than they probably even realized. And yes, it was no small feat to make that whole thing work. We committed to that particular machine a long time ago, and I'm sure there were times along the way where we were probably saying, should we have used a bigger machine or something like that? But we essentially just put our heads down, and we found solutions to all of the stumbling blocks, and we're sitting there with a cell processing protocol that automates this whole thing and does so reliably. That was a critical milestone for the FDA approval process. In other words, we can't submit the IND until we can show that we can do this reproducibly, do it the same way every time. But what's was sort of the magic number that you were looking for here? Because I think that what I heard was that what finally locked the cell processing protocol is we did a pilot run or a couple of pilot runs that showed us that we could get a certain number of HIV CD4 specific HIV specific CD4 cells that we could protect against HIV entry in this blood sample that we could then later restore to patients. So how did that look overall? Like, where did we land in terms of our aspirations on that?
Well, we set up a number of really critical criteria for this process. So it had to be efficient. It had to be able to be done with one leukapheresis pack from a patient, not multiple bleedings. It had to be done with a reasonable amount of the lentivirus vector that we've manufactured so that the cost didn't inflate out of control. And it had to be done in a small number of days. To give you an example of another product, when people are making these CAR T products, which are also leukapheresis packs, then modified with lentivirus vectors to create that form of tumor-killing T cell, those processes are not optimized at all. They take the largest leukopack that you can generate. They take about 28 days of the process in the manufacture, and they take a massive amount of lentivirus vector, about 10 times what we use. And so our process takes a small leukopack one time. It takes 12 days from the leukopack to the time we put the cells into a frozen state to preserve them, to keep them for the patient use. And it uses, as I said, about 1/10 the amount of lentivirus vector that's used in the CAR T process. And we think that these are very important issues, not only now, but down the road. This makes us a much more competitive product down the road because it's got built-in economies and built-in efficiencies already. And to give you an example of really how this is working, we always look back at another company named Sangamo, who did a molecular biology modification of T cells and put them back in. And we ask these cells that Jeff referred to, which are the so-called CD4 T cells that help and drive the entire immune response that's necessary to control HIV. That's really what we're after. And we want those cells back in somebody in high numbers, and we want to protect those cells with our lentivirus vector so HIV can't kill them the next time around. So how many did Sangamo give? So Sangamo would go through a lengthy process, which was about 35 days. They would do leukapheresis and then vector modification of the cells. And they would put back a product into patients which had about 300,000 of these type of cells, but only 1/10 of them were protected from HIV. So 30,000 cells. Remember, your bloodstream is 10 liters. That's only 3,000 cells per liter. A cell is small, a liter is big. So it's a very rare cell. So how many do we put back in? 600 million. So 30,000, 600 million. I'll take the 600 million and be happy with it. Wow. And we'll go forward with that. And this is where our confidence comes from in this product. Because we have attacked the problem right at its core. We have expanded and increased the dose of exactly what we're looking for, and we've done it in a rapid, efficient manner.
Wow, beautifully stated. So, a process that uses 1/10 of the most expensive element for the modification of the cells. That lentiviral vector is one of the things that drives that incredibly high CAR T cost. It's less than half the amount of processing time. So in other words, from when we do a leukopack of the patient to when they get the treatment back is less than half the time of CAR T. The amount of blood we have to draw is way less. And then there is some evidence of clinical response in the Sangamo study that was only returning, did you say 30,000 cells? And we're returning 600 million cells. And this is what gives us our confidence that we're going to get therapeutic effect with our treatment. And what makes you confident that it'll be safe, that we won't get some sort of adverse event? Do you have some thoughts on that?
Well, we have not only put a number of protocols in place, which are more or less standard in the field for determining the safety, that it's not contaminated in any way chemically. There's no chemicals involved in the process, so it's just a biological product. It falls right into the range of the number of human cells that are being put back into people in a whole variety of different types of clinical trials and approved products. The cells, once we modify them with our lentivirus vector, don't behave any abnormally compared to normal T cells, and we don't expect them to do anything unusual in the body. So we've gone through just about everything that was on an exhaustive list of potential pitfalls and crossed them out using laboratory studies or using small animal models that are part of the safety study plan. And all of that has been completed. It's all now wrapping up into the writing and assembly of the investigational new drug application. And last night I spent 4 hours working on the first draft of parts 1 through 3 of the investigator brochure, which is another aspect of this. So we have real-time things in our hand. We're actually working on the IND submission right now and editing and proofing those things. And I think another important point, when you talk about costs, and costs not only of the product, but what did it cost to AGT to do this? We noted in some materials that were distributed by Novartis that they took 3.5 years to develop a cell process, which from our viewpoint is a pretty simple one. We took 8.5 months to get this done in something that's an extremely complicated one. And I think that partly speaks to progress in the field and the fact that we learn from other people's mistakes, but it also speaks to this team and what they can get done when they put their heads down and focus on an important problem.
Yeah. And also that, we really have a expectation that these problems can be solved, and we have a normal standard operating procedure of thinking outside of the box and just looking for solutions as opposed to doing things the same way that everybody else did it. And it really paid off on this. I mean, that is remarkable. 8.5 months to do the cell processing protocol. And he's really, because I've been there through this whole thing, and there's been a huge number of things where we've had to come up with creative ways in order to get rid of cells that we're not interested in there so that we could minimize the amount of lentiviral vector, or even make sure that we stayed inside the size of this relatively small machine. And I mean, it's just remarkable some of the huge things that we dealt with that were dispensed with in a matter of months, when I'm certain that at larger companies it could have been a matter of years that it could have held them up. So you heard it. I mean, just we have incredible confidence that we are aimed at something that's going to be therapeutic. And I already knew the answer to the safety question, of course, before I asked it, because I've seen these guys all sitting around with incredible advisors, clinical people, research advisors, hospital advisors, and that is the number one thing on their mind is how do we make sure that this is safe? The only thing that we absolutely have to avoid is significant adverse events, and we're thinking about that constantly. On the other hand, we're going into the clinic with something that is, tens of thousands of times better than anything that's been done. And there have been a couple of patients that have shown some therapeutic effects even on those old things. So our confidence level is very, very high. I mean, it's really, this is the reason that I'm so excited and so just buoyed by the progress that we've had over this last year. So I really appreciate you and your whole team. Before I let you go, David, another very exciting thing on my mind is the Gamma Delta project. So just to remind everybody, we got our first granted patent is for stimulation of gamma delta T cells that are therapeutic in epithelial solid tumors. And I probably said this to you a few times by now, but we're expecting a second patent to come in soon. So we'll go ahead and put out a press release when that happens. But that covers two major aspects of that gamma delta stimulation. So we've locked it up really nicely in terms of patents. But what does it mean? I mean, what does it translate to in the clinic? I know we're doing a mouse study now, but is there anything coming out of the mouse studies that would give us some sense of how that might work in the future?
To make a cheeky response to your question, what's coming out of the mouse studies are mice that are still alive.
Well, that's always good. I'm sure the mice appreciate that.
Because when you put tumors in them and you don't do anything to stop it, they're not alive for that long. And so we, around this place, we set a very high bar even for our early experiments. We don't want to spend time on approaches to tumor biology or to HIV for that matter. We don't want to spend time on sort of half-baked concepts or things that do a little bit. If you're going to get on the timetable of AGT, the initial experiments have to be more or less home runs. And so we have been doing this now for about 6 or 7 months, doing mouse studies. And I can tell you the first one was an absolute home run because at the end of the study, all of the control animals that received no therapy but received tumors were long since gone. But we had 80% survival in the group that was treated with both our lentivirus vectors and our gamma delta T cells. And this was a stunning result. It surprised everybody that had anything to do with it. And we've gone on now with bigger and better and beefier vectors and more difficult tumors to attack in the mouse. And we keep getting this same result. Everything we have done to improve our lentivirus vectors has done nothing but make them more potent, more specific for the tumor. We do not have those kind of side effects which we would really worry about. I think the only side effect we've seen is in some mice we killed the tumor so fast that the debris from the tumor made people a little bit sick, made the mice a little bit sick. And so that's a problem we can easily control if we were in a human situation. And I think that we're poised to really put this program on the fast track into clinical trials. And the HIV project is amazingly exciting, but what we can do with this and the number of people that this will impact is even bigger than that and even more overwhelming and more compelling.
It's great that we have something to follow on with, and also this HIV asset could really provide a lot of funding for ongoing projects around here, and Gamma Delta gives us a place to expand into a whole bunch of different cancers and accelerate that program, and that's very exciting. And you won't be surprised at all to hear this, but every time I go out on the road now and I talk to scientists and I start describing our Gamma Delta T cell approach, literally I can't even get to the end of it where they just slap themselves on the head and go, "That's brilliant, of course that's going to work." And sure enough, you're proving it out in the mouse studies right now. So I'm looking forward to getting to the point where the HIV project is stabilized and ongoing, and we can really turn our attention and all our guns on that gamma delta. We also have phenylketonuria in the middle there, which is something where a lot of potential strategic partners are actually reaching out to us, interested in that particular disease. So man, just so much great stuff going on. David, I really appreciate you taking this time to get directly with folks and let them know how things are going. It's one thing when I say it, it's another thing when somebody so deeply vetted and experienced in the field can explain to you and exhibit their confidence about what's going on. I think David is an understated guy in general, but he is Mr. Excitement around this company at this point. When I want some good news, I always head into David's office and just ask what's going on. And then I get back out to what I need to do, is just keep raising money. So anyway, and this is what we're spending it on, all this great stuff. So great talking to you, David. Thanks a lot.
Thanks, Jeff.
Thank you for tuning in, and hopefully we'll see you again soon.